Live from New York, it's Saturday Night TWiV.
Micro TV. This is TWiV, This Week in Virology, episode number 1000. Recorded on April 15th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Thank you so much for joining us to celebrate this amazing milestone, 1,000 episodes of a science podcast. I don't think there are many TV shows with episodes. I hope you've been enjoying the jazz of the Randall Dayton. Alto saxophone, Randall Desplombier. On piano, Randy Ingram. On bass, Aaron Holtus. Rodrigo Recabale. So in the summer of 2008, I had an idea to start a science podcast to record the kinds of conversations that we have that no one else hears. I didn't think it would work. Like most experiments we do, most of them don't work. I didn't think that one would. But it did, and people started listening, and more people started listening. So I started doing more and more podcasts. I started a parasitism podcast, microbiology, evolution, immunology, neuroscience, and infectious diseases. And I was just amazed at how many people were listening. And then, of course, came COVID-19, and TWiV exploded with so many new people, including many of you, joining us to listen. And that gave us the courage to start a production company, Micro TV, to record podcasts in a studio right here on 7th Avenue, not far from this theater. And today we are celebrating 1,000 episodes of one of those shows, This Week in Virology. Just, isn't it amazing how far viruses can take you? So tonight I would like to celebrate all of the individuals. Join me in celebrating all of the individuals who have participated in this podcast over the years. I'm going to introduce all of them to you. Now, back in September 2008, I decided to have one of my Columbia University colleagues join me in that first podcast, TWiV number 1. He's still with us today from Fort Lee, New Jersey, Dixon Desplombier. Dixon, you remember the first episode? You remember how we got to that?
Don't have a clue. Actually, it was a turning point in my life, believe it or not, because I realized how serious it was about continuing to TWiV too. So the first time we did it in my office, and should I tell? Well, it went very well. So, we did a dry run first because we sort of forgot to turn on the recorder. So we went back and did it again, corrected that mistake, never made that one again. We made a lot of others, but we're proud of those mistakes because you learn every time you make one. It's just like doing research. And we're proud of the fact that we could attract so many different people to our subject of virology, and I'm not a virologist by training, but I think I am now.
Yes, we know you're not. That's okay, you're still with us. And I picked you because you're a great teacher, and I thought you would be the one to join me. But it became clear rather quickly that you had other things to do as well.
Yep.
And so I thought maybe we should get some other people involved. And that's where the next co-host came from Western Massachusetts, Alan Dove.
Yay! Woo!
Good to be here.
Those words. Do you remember how you joined TWiV?
I do. It was, I guess it was about 2008, and Vincent and I have been in touch over the years since I'd done my thesis in his lab, and occasionally I'd hear from him. And he sent me an email and said he'd started a podcast. And would I take a listen to it, and what did I think of it? And being a journalist, I had kind of followed the whole podcasting trend, which was just getting going then. And I listened to the first couple of episodes of TWiV, and I wrote back to Vincent. I said, this is brilliant. This is a perfect use of this narrow casting medium, because there are maybe, I didn't tell him quite this way, but I thought there are maybe 1,000 people in the world who will listen to this ever. But you can reach that kind of an audience, that kind of a global audience, with a podcast, and that's perfect. And then he said, hey, why don't you come on the show? Oh, okay. So I think I was on as a guest talking about science journalism one episode, and then he never got rid of me.
No, I think having a writer was the idea that we would have a different perspective. To this day, you provide it, so we're very grateful. And the next host we added to the show from Austin, Texas, Rich Condon. Can't handle your adulation. So, Rich, how did you get onto this book? Do you remember?
Yeah, so first of all, importantly, Vincent and I knew each other from serving on an NIH grant panel, review panel together in the early 90s. And 26 episodes in, you were starting to do individual virus families. You wanted to do pox viruses, you knew. I worked on pox viruses, you knew from the grant panel that I could talk in complete sentences. So you had me come on and do that. And then I looked up the old episodes for the next, over the next 11 months. I came back for 6 other sort of guest episodes until finally you said, hey, dude, you know, why don't you come on for good? And that was episode 68, Ode to a Plaque, still my favorite.
Yeah, Rich said originally, I'll pitch in when you need me. I said, no, we need you all the time. All right, and then next to join us from Ann Arbor, Michigan, Kathy Spindler. Kathy, you remember how you joined?
Yes, I remember, first of all, Rich and you came to Michigan, and we did the TWiV in November of 2011. And then you asked me to do a couple of episodes, because nobody else could do it. So I was sort of an ad hoc standby or something. And then you invited me to join in August of 2012. It was just before you were doing the TWiV 200, and you'd already made the plans for that, and you couldn't squeeze me into that. So.
I think it's important to find people who really want to do this, right? Not everyone wants to commit the time. So you give them a little dry run and you can tell. Not everyone makes the cut. This is a great team that I've built here, and I'm so proud of them. Next to the list of TWiV hosts, from Madison, New Jersey, Brianne Barker. remember when you came to TWiV?
I did. I met you at a conference and we started talking about TWiV, and you mentioned I should stop by if I was ever in New York. So I stopped by for episode 432 in March 2017. And then you were looking for someone else to replace some hosts when people were absent as a sub, and I volunteered. I did that for about a year or so, and then around episode 482, March 2018, I joined after that.
Isn't it cool that everybody remembers their episode?
People might have known this question was coming in.
Maybe. Our episode's easy.
With my memory, number one, after that it's a crapshoot.
So our next host was clearly a result of COVID-19 pandemic from New York, Daniel Griffin. You notice his bow tie, there's no viruses on it. Right?
This is a special occasion. You're seeing me maybe one of the few times in my life without a virus-inspired bow tie.
So Daniel,
You can get pictures later.
do you remember the circumstances of coming onto TWiV?
I do actually. I was thinking about this just before I came in because there was sort of like a couple of little parts. One was sort of that step in the water, and the other was a jumping all the way in. So it was February 2020. And I came by because I was already thinking that something big was about to happen, sort of saying goodbye to everyone, saying, you may not see me in the lab for a little bit. And we did a special little episode with Ori, if you remember.
Yeah, I do.
And that was my usual, I don't know, socially distanced self. I'll do the episode, but everyone has to face different directions and sit 6 feet apart. And that was the first sort of step in the water. And then, a little bit afterwards, I called up Vincent. I said, Vincent, we should do a once-a-week clinical update, a COVID update for clinicians, so clinicians are up to speed on what's going on, what we're seeing, and how we treat COVID. And wow, 3 years later.
Yeah, we're still going. For a while, we did 3 episodes a week.
Yes, I was only invited to 1 a week.
You were saving the world, man.
We were busy.
All right, and our final host from Montreal, Canada, Angela Mingarelli. So Angela, you remember how this went?
Yes, I do. We met in July 2022, last summer. I was at an infectious disease symposium for bad viruses, well, bad pathogens, and during one of the breakfasts, I saw Vincent, and I walked up to him, and I was like, I am such a fan of TWiV. I love TWiV. And we started talking, and I told him that I was a PhD student, and that I was also a veterinarian, and he said that, well, we need a vet on TWiV, so let's see how it goes. And I started TWiV episode 946 was the first one, Flu versus Flu, and here I am.
I have to say, it's not easy for a student to walk up to someone and just say, hey, I like what you're doing. It's tough, right?
Mm-hmm.
So I thought that in itself was a good sign. And so Angela's doing once a month because she's a PhD student, but maybe afterwards that'll change.
Yeah, we'll see.
So that is our team, folks, and we'd like to chat a little more amongst ourselves. And by the way, we're going to talk here for about 15 minutes, and then we're going to ask you if you have any questions or comments. We'll have a microphone being walked around. And you have Daniel here. You can ask him if you need that latest booster or whatever. But first, I wanted to ask each of you to tell the audience a science story that means a lot to you. So, 5 minutes, Dixon.
All right, so my training is in parasitic diseases, and I taught at Columbia for a long time. The second year medical students, as a lot of you know, just by listening to the shows. And my interest in viruses were more of the vector variety than of the virus itself. And I've had a lot of courses in medical entomology and stuff like this. So when I heard about This Week in Virology, I said, But I knew the person who actually discovered Lacifina. I actually sat next to him in our offices when I was a graduate student. And so it was really interesting to see the evolution, how that evolved into, first of all, the biosafety hood, and then the biosafety 2, biosafety 3, and biosafety 4 laboratories. And that's all evolved since the discovery of that virus. That was back in 1967. Then I decided, and all of a sudden in 1999,
Dixon, they're trying to get you to look at the camera.
I'm sorry. Who's disagreeing with me? Then, in 1999, the summer of 1999, the first cases of West Nile virus occurred in New York City. And that's when I really got interested, because I had a medical student who wanted to work on that, because she also wondered where did it come from and where is it going, basically. And so we set the groundwork up and got all the papers on West Nile virus together, perused the literature. Then I decided to write a book about this. It's something, it's called West Nile Story. It's sort of a takeoff on West Side Story.
I need you
I
to
will
sign
that.
this.
I will sign that.
I need you to sign
I
this
will.
after.
So, yeah. So guess, if you read in the beginning the acknowledgments in the help, this gentleman right over here rescued me from viral ignorance by simply reading the text and correcting all the mistakes that I made. And at that point we became friends. I started paying more attention to viruses as parasites, or at least as parasitic organisms. And from then it was infectious, even though I didn't take This Week in Virology as seriously as Vincent did, because of course I didn't start it. I didn't realize how adamant and how persistent and how wonderful he really was. I have a deep, deep abiding respect for all the work he's put into this. Every weekend he sits and edits everything. And it's amazing the amount of work he puts in, and it's all out Sunday morning.
Okay, time's up.
The check is in the mail, did you say?
Okay, so what is the mantra that I am supposed to do with Dixon?
Be nice, yes.
Have I been nice?
He's been wonderful.
All right. Alan, tell us a story.
So I thought about this, and I realized I've got a great story from Vincent's lab. I'd been in the lab for about 2 years. I was working on a project where I would purify a large quantity of the poliovirus receptor, the cell protein that gets it into cells, and send it to our collaborators at Harvard so that they could crystallize it and get the 3D structure of it. Very cool project. In the couple of years that I worked on it, I made the singular discovery that I am not a protein chemist. So I stopped trying to be one, and I came up with a new project which was looking at the early entry steps of poliovirus getting into cells. And at that time, nobody had done a really detailed analysis stepwise of how that worked. So we knew the virus bound to the receptor, which was discovered in Vincent's lab, and then it could undergo this major structural change called alteration, creative, I know, where the capsid spits out one of its proteins and changes its shape. And the thinking was, okay, that's kind of a loosening of the capsid so it can uncode into the cell. But we didn't even know if that was the correct pathway, because most of the viral particles with polio don't succeed in initiating an infection. So this could have been a dead end, or it could have been a productive step. And I thought, all right, we know that this alteration process only occurs at temperatures above 33 degrees C. And how about if we just see if the virus can infect cells at 25 degrees C, room temperature? Great, Vincent said, sure, go ahead. Well, room temperature, of course, fluctuates a lot, especially in the Hammer building, which had a horrible HVAC system. So first thing I had to do was build an incubator that would maintain 25 degrees C, which is harder than it sounds. I finally got that going. And I did the first, most obvious experiment, a one-step growth curve, which if you've listened to Vincent's Virology 101, you know this means you take a plate of cells, you put on 10 times as many infectious units of virus as you have cells. That ensures all the cells get infected at once, and you get this nice, very rapid, or in accordance with the speed of the virus, you get a curve that shows you the viral replication kinetics. And I thought, all right, let's do that at 25 degrees and see what happens. Either I'll get a one-step growth curve, probably delayed because it's colder, or I'll get nothing. If the altered particles are required and they can't happen at 25, it could be a flat line. So I did that, and I did my plaque assays after I'd taken time points every 12 hours for about a week to give it plenty of time to replicate. And I did plaque assays on all that, and I plotted it, and what I saw was neither of the two expected results. I got replication, but it was not a one-step growth curve. It was this long lag, and it finally picked up at the end. And I realized what had happened was the wild-type virus indeed can't replicate at 25 degrees, but I had some rare mutants in the population that could. And I got this, result, and I graphed it. I said, wow, that's really cool. I just found something new. I've been in the lab for almost 3 years. I finally found something new. So that is a really awesome experience in science. And what made it even more cool, for me at least, was I had done this with essentially 1950s technology, which underscored that you don't always need the cleverest experimental design or the latest technology to find something new, even in a system that's been heavily studied. And I thank you for enabling that opportunity.
And then you went on to be a science writer.
Then I went on to become a science writer because I said, all right, I had that rush, not going to chase that rush. See ya.
Rich, what's your story?
So the charge here is a transformational story, and I keyed in on the word transformational, and when I thought about that, this story just popped into my mind, so that means it's transformational. I was a first-year graduate student, so we're 1971. I had just joined Joan Stites' lab. She is a scientist of some significant repute at the time. However, she was a new assistant professor, 30s something. There were just the two of us and the technician in the lab. We had these bays that were sort of islands in the lab, and she had a bench on one side, my bench was on the other side. She worked in the lab all day long, I worked in the lab all day long, and we were just there doing our stuff. And new graduate student, I asked her about 1,000 questions a day, okay? How do you do this? How do you do that? What does this mean? What would be the better way to do this? How many of this do I need? Blah, 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 blah. One day, and I'll never forget, if there are scientists of my vintage in the audience, some of this will be familiar. We didn't have Appendorf tubes. We didn't have pipetting. We had little glass micropipettes that you mouth pipetted with, all right? And we had 15 mil Corex tubes that had to be siliconized because you spin down the RNA into a little pellet in the bottom, and then you could pipette in like 50 microliters of fluid, and it rolled around so nicely on the siliconized tube, and you'd be looking at the pellet to resuspend it. Joan was doing this. She was in her coat. She had her 15 mil Corex tube. She was looking up. When you did this, you'd have to look at it really closely, and your eyes would cross, and I can still see her doing this. I went up, and I asked her one more question, and she gave me the dirtiest look I've ever seen. And she said, "Go figure it out yourself." So that was my first explicit lesson in critical thinking. And I think that's what this is all about, more than anything else. The discipline, TWiV, everything else is about critical thinking. So thank you, Joan. Thank you, Vince.
That's great. Love it. Kathy.
So ours is a business of delayed gratification. And so when we were supposed to think of some transformative thing, all I could think of was whenever I got a result, I was never sure of it. We had to do more experiments, so I could never really pinpoint things. But so looking in retrospect, think about sort of the milestones for me. And the first was my first published paper, which was on Phi-X 174 when I was a graduate student with Masaki Hayashi. And he and I were the only authors of that paper. My next recollection of something was when I was postdoc at UCLA, and when I got my first paper there, which was a really long haul because I had to learn how to do organic peptide synthesis with C14 labeled amino acids, and it was awful. But we got an antibody to the adenovirus E1A protein, both through that and through a cloning approach, and I basically had to learn cloning. So that was it was a long haul, but it felt successful. When I moved to the University of Georgia with my own lab, one of the big milestones for me, again, was one of the first papers, but this was a paper where the first author was one of my trainees, and so that, for me, was really rewarding. I have a couple of other things, milestones, I guess. So one of the things was when we made our first mouse adenovirus mutant. That was at Georgia, and that was using pretty old technology, but that was really exciting. But again, it wasn't just one particular day. It was, well, we think we've got the mutant, but now we have to do these additional experiments, and so that delayed gratification came in. I moved to the University of Michigan, and the milestones there, I think, were when we started doing the genetics of susceptibility to mouse adenovirus, and when we were able to publish a paper on that was really exciting. And we also did work on the blood-brain barrier, and these were two areas of science that I never envisioned going into. So it's another aspect of science is that you go where the science takes you, not where you think that your best skills or training are. And so I wanted to say one thing I forgot to say earlier about, so another thing that happened in Michigan is when we had the TWiV come there in 2011. And it was, for me, I was helping to organize it, and then Vincent said, oh, Kathy, I want you to be one of the guests. So then I had to switch, and I didn't publicize it as much as I should have because I felt like I was publicizing myself. But for me, it was like taking my orals in front of my entire department. My oral examiners, the department chair were there. There were about 4 or 5 other faculty members there. And Rich and Vincent, in their calm way, kept asking really good questions, most of which I had to say, well, we don't know the answer to that. We're working on it. So that was fun. I also have a mentor story, and that is that I changed advisors 4 years into my PhD. Some of you have heard me talk about that before. And I started working with John Hawley. But I particularly remember that I had talked to him and a couple other people, and he was the one who was the most welcoming, and I decided I would go work in his lab. I had not done a rotation there. So he was taking me without knowing anything other than I had a paper on 5X174. And the day that he took me into the lab was the day that he learned that someone had cross-threaded one of the fixed-angle titanium ultracentrifuge rotors, ruining it, making it basically a brick. And I don't know how many thousands of dollars these things cost then, but he just calmly said, well, that's that, and just, what can we do next? And he said, yes, Kathy, I'll take you into my lab. And that changed my life, so I'm really happy that the centrifuge rotor didn't completely ruin his day.
That is important. Another way to put it is what it is, right? Okay, Brianne, how do you?
When I heard this question, I, like Rich, really focused on the transformative part of the story. I was an undergraduate working on HIV vaccines and specifically thinking about aspects of adjuvants, and I went on to work in a lab in graduate school that worked on those same types of questions in some non-human primate models. I was particularly looking at CD8 T cells and how we could use cytokines or adjuvants to influence the CD8 memory response. And at one point I went into my PhD advisor's office and I told him about some of my ideas for what I might do next. And he took one look at my list that I had given him, and he shook his head and he said, no, absolutely not. You cannot do any of the things that are on this list. If you do any of the things that are on this list, you will be training to become Norm the Second. And the world doesn't need Norm the Second. The world needs Breathe First. So I want you to go back to your bench, and I want you to start thinking about what unique things that you can bring to science and how you can put together some new skills that no one else had. I started reading a lot more broadly and got very fascinated by the idea of how certain cytokines that were coming from the innate immune response influenced viral pathogenesis in non-human primates in the SIV model. And I wanted to learn as much as I possibly could about how those innate immune cytokines were made and what might influence that. So that made me switch gears entirely to the innate immune side, where I went for a postdoc and what I now work on in my own lab. And I often tell my students that they need to think about what's going to make them themselves the first in going forward with their projects.
Nice, I like it. Breathe first. All right. Daniel.
All right, well, for starters, hello, everyone. I might need a little audience help on this because I think the question was like, which virus? But here's the first question. Can my story involve more than one virus? Thank you. So we'll go back. Here I am, high school. It's early 80s, so I've already dated myself. My mom's a painter. We're living in Soho. We move over to Greenwich Village. Early 80s, I'm in high school. I mean, I've heard all about, the free love of the 60s. I'm excited. But our neighborhood starts undergoing a change. From this vibrant neighborhood in the early 80s in Greenwich Village, all the people are out late, all the exciting happenings, suddenly something starts to transform our community. And if people remember, this is when HIV first gets introduced to New York. And in a matter of a few years, our neighborhood completely transforms. It goes from this vibrant, exciting, to really sort of a frightened neighborhood, a frightened community. So this is my first sort of interaction, and it's not a good interaction. I'm volunteering at St. Vincent because no one will want to. Where should I be looking? Should I look at you? Okay. Just keep talking. So I'm volunteering at St. Vincent's Hospital because no one really wants to transport my neighbors. Everyone is living in a sense of fear. So I go off to college. At this point, I'm not excited about medicine. This, if anything, viruses are just a horrible thing that has just really devastated my community. So I don't go into medicine. I avoid the sciences. I end up as a philosophy major. I end up actually in 1988 headed to Kunming, China to teach in the philosophy department at Kunming University. All right, I'm set. I know what I'm going to do with my life. I'm heading up into China, and I get sick. I get a headache. I get this pain behind my eyes. I get so sick, I realize I kind of need to get home. This is pretty bad. I end up home in the States. I get this horrible rash, this red rash, sort of white islands on a sea of red. I feel like my bones all hurt. I go to the hospital because, I'm young. I'll get better, and so I think that happens. But now it sort of interrupts that trajectory, and I end up, a few years later, now I'm in medical school. I'm interested in infectious disease. I'm not interested in those viruses. I'm interested in tuberculosis. That's a good disease. And now I go off to Africa. I travel again. I haven't learned my lesson. So I spend a couple weeks in Africa. I'm canoeing down a river. I'm hiking through the Svelte. Is that the right word, Dixon? The Svelte. I'm staying.
No, your spell. You were.
Okay.
It's a V, it's a V.
Okay, so the place with the V. I'm staying in pop tents because I'm under the impression that the hyenas, the lions, etc. will not enter a pop tent. But I come back, and again, on the way back, I start to not feel well. I start to get this fever, a bad headache, that pain behind the eyes. This time it's worse. I feel like someone has broken every bone in my body. And so, it's late, I get back, it's pretty late. I don't want to bother anyone, I'm trying to be considerate. So I write out my proper H&P, just worried that in the morning I may not be coherent, properly predicted. End up in the hospital. Again, I just feel like someone has broken every bone in my body. Again, with the rash, my platelets drop down into the single digits. And we think this is my second experience with dengue. Now I'm hooked on viruses. Right? So about 10 years ago, after completing my MD, my training, my infectious disease fellowship, my PhD, I come to Columbia, and my agreement with Scott Hammer is I will only work on HIV, I will only treat HIV patients. I am now full gone with the viruses. And then as we know, in the end of 2019, early 2020, everything changes, and the last virus to mess with my career plans has been SARS-CoV-2.
Is it the last?
I hope so, but I do not think it is.
Okay, Angela, what about you?
Okay, I'm the last one. So my story is a little bit less transformative and more just a viral experience, my first viral experience, let's say. So I started in my lab in 2021, in the winter of 2021. and fresh out of vet school, a few months. I didn't really have much viral experience in the sense that I had learned about viruses in vet school, all in different animals, and wild, domestic, but I had never actually propagated a virus. I barely knew what a plaque assay was. So when the pandemic started, well, this was mid-pandemic, my supervisor got interested in T-cell responses in the human endemic coronaviruses, so HKU1, 229E, NL63, NLC41, which I'm sure many of you here have heard about because we talk about them all the time on TWiV. So I'm like, okay, I get a protocol and I propagate beautifully 229E in Hu7 cells. It worked so well. I got so cocky. I was like, oh yeah, 3.4 times 10 to the 7 titer. Oh, I got this. So then next virus, I was like, okay, I have NL63. I'm a pro. It's fine. I've done this once. And so I tried during 6 months in multiple cell lines to propagate this virus, and I was already working with very low titers, and it just didn't. I couldn't even get a signal via qPCR. Like there was like no virus, and we tried so many things. So I leave the project I leave the project altogether, as we do in science. Something doesn't work. You eventually reach a point where you have to move on. I move on to other things, and about 4 months later, I'm part of a virome effort. So somebody was doing a virome study, which is basically studying the viruses of the upper respiratory tract, and I was a healthy control. So I get swabbed, and 2 days later I fall very ill. So I had a fever, I was at home, and I was like, oh, I just got virome sequencing, I wonder what it is. I guess we'll find out in a month. Everyone in my lab thought it was SARS-CoV-2, but I had a negative antigen test, and also PCR, and then we thought it was RSV. So I was like, oh, it's probably RSV, we'll see. A month later, I get the results back, and I see the results. Top viral hit on the sequencing, coronavirus NL63, and I was like, yeah, of course it is. I tried to grow you in flasks for 6 months, and this virus is just shedding everywhere, millions of virions, and it was just such a moment of like, of course it is. Yeah. And now it's like an ongoing joke on the floor. They're like, oh, you're going to try again? I was like, no. That was my transformative experience.
So some people do experiments and some people get infected.
Yeah, it wasn't my virus because I was working with very low titers, and this was months later in case people thought definitely not.
Do I get to go?
Yeah.
Yes.
All right, so I think I'll go back to the beginning, and many of you may have heard this, but I went to college, and my dad was a doctor. Of course, wanted me to be a doctor, but I didn't want to, actually. Didn't have any appeal whatsoever. And even after I visited a resident in a hospital, and he talked to me for a while, and he took out a tube of KY jelly, and he said, I have to go do an exam, a rectal exam on a 99-year-old patient. And I said, nope, that is not. So I get to Cornell, and I'm not sure what it is that I want to do. I didn't find anything. I was a biology major, so I took a lot of science. I loved science. I had a great high school science teacher who put that in me. So I said, what am I going to do? I go home, I go back to living with my parents. I get a job as a lab tech doing some microbiology. I really, really enjoyed that. And then one day, I read this book called Fever, which Dixon is aware of, by John Fuller, an account of the emergence of Lassa virus in the 60s, one of the first, if not the first, emerging virus that we know about. And I say, oh my gosh, viruses can do this? I need to do these things. But how do I do it? Because I don't have an advisor anymore. I'm at home. So I started applying to master's programs here in the area. And then one night, this is the transformative night, I'd gone to school with a young man named Ed Kilborn. And his dad was also Ed Kilborn, or I should say Edwin. And he was chair of microbiology at Mount Sinai. So he lived in Ridgewood, not far from where my parents lived. So I went to his house for dinner one night. And after dinner, Dr. Ed Sr. came out and said, so, Vinny, what are you going to do with your life? And I said, wow, he's interested in what I'm going to do. That's interesting. I said, well, I want to be a virologist, but I have no idea how to do that. And he said, well, come to Mount Sinai and get a PhD. And it was August, as you know, applications are well over. I went for an interview the next week, and I got in, and I started in the program. And that's why I am here today, because he gave me good advice. And so The lesson for me was find people to listen to. It's lesson number one, and listen to them. It's all about mentoring. And the second is there's no linear path. You can wander around. You don't have to have it all planned out. I tell that to students now, and they're amazed. They say, "What? I don't have to have all this planned out for the team." No, you can just experiment and try things, and often that's the way to do it. So I'll never forget that story. And I wrote a blog post about Dr. Kilbourne's influence on me many, many years ago. When he passed away, his family asked me to do a eulogy at his funeral because I had written such nice things about him. But they were all true. So that is my story. Now, I would like to hear from some of you who have questions. There should be a microphone ready to carry around, right? Grab that one. Well, who's going to carry it around? Where's it on the podium?
Take it off.
Oh, give it that. Where's Nadia? Is she here? No.
Nadia.
She's in the, oh, give it to Amy. She'll take it around. Okay.
Wait,
so,
he's got a TWiV shirt.
he's got
a TWiV shirt.
I know he's leaving. Is that enough?
So to give you time to incubate, where are you from? Shout it out.
Ohio.
California? California's the farthest, right?
Louisiana.
Louisiana? Wow. You guys came all the way to see this? It's impressive. There's a question over there.
Yeah, but this man was first.
Okay.
But
It's not working? Can
We
you
don't
show
have
me?
working microphones, so you'll
No,
need to
maybe the
project.
switch.
Sure, I can show. Do we know, or is it one of the big open questions in virology, why there's such a huge range in the durability of an immune response? Some viruses, like influenza, mutate, so the response lasts only a couple of years. Others, like polio or formerly smallpox, you get one exposure and you're protected for life. Why are some viruses able to do that and others can't? Do we know? Big picture principles.
Everybody looks at this analysis. We do this on the show too, as you know.
I would say that it is definitely an open question. I think that some of the details have to do with the amount of viral replication and the locations of replication, as well as ways that the viruses might be evading immune responses or manipulating immune responses. But overall, I think that there's a lot we still need to learn about that.
One of the ideas which was espoused by our microphone carrier, Amy, is that viruses with a viremic phase, where the virus is in the blood, they have access to more lymph nodes,
Exactly.
right? So they can make a broader, more durable response, whereas the mucosal infections are limited. That makes sense.
That does make sense.
But you need to really test it. It's kind of anecdotal
Right.
association.
It's truthy.
It's also kind of hard to find one virus that you could use for both a viremic infection and a mucosal infection.
Measles,
Good
right?
point.
The mucosal infection, the viremic stage, really good immunity, durable immunity, right? I haven't heard truthy in a long time. That was at the beginning of the pandemic, yeah.
Would the viremia mean it's also got more chances to get picked up sooner, or is it just different kinds of responses?
I think the viremia is just about accessing more lymph tissues, right? Instead of just lymph tissues in your neck, it's going to be throughout your body. Okay, there was one here, right?
Thank you. My name is Siddhartha. I'm from New York.
All right.
My question is this. Initially there was an idea that the virus wouldn't mutate much because it has this proofreading mechanism. And then we have so many mutations and so many changes. So I was wondering, does that challenge our knowledge, or is it just because like the virus had such a bigger population, and or was there an example where there was some virus with a proofreading mechanism that didn't mutate much, and this one was a surprise?
Well, I think even with the proofreading, exonuclease, it's still pretty high mutation rate. Maybe tenfold lower than other RNA viruses, but it's quite high. But I think the kicker is, once you get into millions and millions and millions of people, then you're going to get a lot of mutations.
And animals.
And animals. Remember the first months of the pandemic, we hardly saw any changes in the genome. And then once it got into tens of millions of people, boom. So it's all a numbers game. So I don't think it's teaching us anything new. So it makes sense?
I would agree with that. And I would say, looking at the other common cold coronaviruses, you see genetic mutation happening all the time with those, and they circulate at similar levels. People just don't pay attention to them. So yeah, they all have proofreading mechanisms too, right?
That's right.
And they all mutate because that's what they do. But this is pretty much the pattern we ought to have expected, whether we did or not.
One of the, I can actually say something here because I know all about it. The oldest vaccine that we have, correct me if I'm wrong, is the yellow fever vaccine. And that has remained steady. Smallpox, that was a scarification, but I'm talking about an injectable vaccine that you can get
Keep trying.
right now. No. So is it the same as it was in the very beginning and now? Because it still works. And it's still the same vaccine again and again. As the mutation of the virus vaccine, it's a COVID vaccine, has not escaped from the normal immune responses that people give to it. So that's the opposite end of the spectrum that you're looking at. You got the COVID's over here, and you got the yellow fever's over here, and everything else.
We're using the same polio vaccine.
See, so I did learn something.
Polio mutates like crazy, but there's a constraint on it that prevents it from becoming too antigenically different from the vaccine.
So, another yes.
Yeah, serious question. Did you have any idea when you started TWiV how many people you would reach and the interest that it would generate?
Yeah, I didn't. As I said, I thought it would fail. I thought no one would listen, right? We're going to listen to a couple of scientists talking about viruses.
I work in the media industry, and I thought this had a global audience of maybe 1,000 people. And what shocked me, just completely blew me away, was after we'd gotten a certain distance into the show and we were getting emails from people, and a lot of the emails were from the people who I thought were our core audience anyway, the postdocs, the graduate students, faculty members occasionally. And then we were getting emails from, like, I'm a long-distance truck driver, and I listen to your podcast. Wait a second. There are people out there who don't work on this for a living, who are just interested in it? And
Ask us how many. In this room.
Who are not scientists? Raise your hand.
First, away from the studio TWiV show, TWiV, what was that?
34?
It was in Philadelphia. We had 4 people in the audience.
One.
No. Counted us as part of the audience. Vincent, myself, the cameraman, and one person.
I was there.
You were there.
There were 5 people in the room. Yes.
So the question should have been, why didn't we quit while we were still behind by a little bit? And we had faith in this to the point where we knew that somebody was listening. They had to be.
I just enjoyed doing the show. I don't know. You had faith in it, but
I had faith in him. I had no idea where this was going. No idea.
I want to make 2 points here. One is, one of the reasons why I ended up writing to the show before I became a co-host was because you oftentimes have to fact check Gibson. So, the second is that for those of you who won't be able to see this, about half the audience here raised their hand as non-scientists.
And while we're on this subject, so I'm amazed that this room is full, right? This just blows me away that you guys came to see this. My favorite comment was from a policewoman. I don't remember where she is. Beginning of the pandemic, she wanted to know about PPE. She said, my only PPE is latex gloves.
Non-latex gloves.
Non-latex gloves and a Glock.
Not a clock.
We often would talking. And then a year later, she wrote back and she said, yeah, I'm still here, it's Cindy, and she had some other questions. So that kind of thing is amazing. Any others?
Thank you. So I have a science question. As much as I love viruses, I'm very interested in the idea of making certain viruses extinct. My question is regarding polio vaccines. It seems clear that neither OPV or IPV is feasible for eradicating polio. Is that fair to say? As they are now. Are you aware of research going on around making IPV give mucosal protection so that it could be used to give gut immunity? Maybe testing different adjuvants or things like that.
I see Amy is dying to answer
So technically, I'm not allowed to answer your question as a representative of the FDA. I will be happy to discuss it. I do not wish to be fired.
The answer is there's a lot to discuss.
There's
That is an excellent question.
The idea of eradication is incorrect.
Okay, go ahead.
Who's keeping an eye on bird flu for us? The avian influenza is not sounding good at this point
No,
to me.
it's not sounding good at all.
Yeah, so I'm worried, but I'm not a scientist. So are you guys worried?
Well, we had an influenza expert on, what's his name?
Scott Hensley.
Scott Hensley, and I asked him, are you worried about H5N1? He said, yeah, the world is not ready for another pandemic. That is true, right? The concerning aspect of H5N1 now is that it's starting to infect mammals in a big way. Before it was just a human here and there, less than 1,000 infections, but now it's infecting all kinds of animals, and that will allow more mutations to be selected. So I think we need to be ready. We have antivirals, we have several that work, and we need to make some vaccines and test them quickly. So we're not caught short as we were for this one, because we shouldn't have been. So
Yeah, I
okay,
think also, I'll say sort of positive. I do think certain things were learned during this pandemic. I know there's a lot of, like, we messed up a lot, just to be straight. But like the monoclonal story, for instance, that was almost a disaster, right? When we first got involved, it was early April, and I remember a call with Regeneron, and what are you guys doing testing monoclonals in people who've been in the ICU for 2 or 3 weeks? They're not not going to work, but then no one's ever going to go near these things. And so, one of the big things I think we've done is we now have set up relationships where we can test things during the first week. And that's when you want therapeutics tested, not once you're already in the ICU on a ventilator. So I know there's a lot of like, oh, we've learned nothing, but I'm not as pessimistic. I think we've learned quite a bit, and particularly with H5N1, I feel like we'll be in a better situation. But I am worried about it, by the way.
Just to add, I think that it's also, we've learned also with SARS-CoV-2, it would be important to continue surveying animal reservoirs, potential animal reservoirs, because it's in other mammalian species, just to be testing them to see, especially near city centers and things like that. It'll definitely be important, which we are now doing as well with SARS-CoV-2. So,
Amy, you can let Nadia take the mic. Have a seat. Thanks.
So, especially thinking about coming pandemics, how are we to deal with the current disparagement of knowledge and the denial of scientific fact that is happening in our societies? But these are very, it's very easy for us to sneer at people who say that scientists know nothing. But that is a real force. It's a real presence in our society, and it's permeated deep into the politics, not only of this, particularly of this country, but not only this country. What are we to do?
Well, I think that's an area where we have also learned a lot, and I say we collectively, the media industry, and also scientists and public health officials. When we had Laurie Garrett on the show, she commented about something that I had also observed, which is in these meetings that have been going on for decades, where people talk about what do we do about the next pandemic that everybody in infectious disease knew was coming, and my money personally was on it's going to be flu. Okay, so I shouldn't place bets. But in those discussions, there were like ongoing things, tabletop exercises, scenario planning, and in none of those discussions had anybody contemplated things like heads of state promoting disinformation to their own people. That was just like not in the conversation. It is now. So hopefully we will be better prepared for that, but there are limits to what we can do, especially in the age of social media that is driven not by fact, but by attention. And if you're scrolling, then they're winning. And you will continue scrolling when it's disinformation because it made you look, didn't it? So I don't have a very optimistic answer about that, but I will say that the people who should be trying to deal with it are now more aware of the problem than they used to be. And hopefully we will do better on that score as well.
The one thing that I would really like to add is that I've learned and realized during the pandemic a lot from many of the people who I've spoken to. I'm from a very, very small town. Many of the people who I grew up with are not people who spend a lot of time thinking about science, and they frequently contact me to ask me questions, and they talk about how they know me and so they trust me with getting information from me as opposed to a lot of different outside groups. I learned about a group from Vincent called Research America who asks a survey of American adults every year, and one of their questions that they ask is how many of these people can name a living scientist. In most years, a maximum of about 20% of people can name a living scientist. What that says to me is that not only do those people probably not naming a scientist, they probably don't know one who they can ask questions to, like some of my friends and family have during the pandemic. So I also think that there's a bit of an impetus on us as scientists to be more public and to talk to people and help people understand that we are people like them, and that we have good intentions as we are doing many of these things, instead of being the boogeyman that sometimes people imagine a scientist to be.
I think there's someone here with the mic, right? Yeah.
Hi, this is Masa.
Hi.
First, I'd like to thank you for taking time and sharing your knowledge with us. Appreciate it. Last week, I attended the World Vaccine Congress in DC, and apparently pharma companies are trying to convince us that they are working on vaccines that prevent transmission and infection. But we know that vaccines are merely prevent the disease and hospitalization. Do you think is it at all possible to make these kind of vaccines that prevent transmission?
So we talk about this all the time, right? The only way is to have high levels of mucosal antibodies for extended periods, right? Which physiologically doesn't happen. So I don't think you can do it. Now, I would hate to tell someone not to try something, right? Because that's anti-science, because that's how we do things. So who knows, maybe someone could come up with something where maybe, is this even possible? Memory responses are triggered really much faster? Probably not.
But it may not be something we want to mess with, though.
Yes, so I have problems with that because, natural infection has historically, throughout long-term evolution, prevented disease, and that keeps a species going. That's sufficient. So it's not clear to me why we want to do that in the first place.
But it doesn't have to be perfect, right? It could just reduce the risk.
Yeah, you can reduce transmission, but again, you reduce transmission only when you have high antibody levels. When the antibody levels contract, as we know, we have memory to take care of that. That's not a big deal. Let's take one more because we have something else for you. Is there? There we go, back there.
We also have two questions.
Okay, let's exhaust them and then we'll move on.
Oh, okay, Charles from North Carolina. Very light subject. Your favorite off-virus subject that you get on manual transmissions, stump grinders, snow shovels?
Oh, what's our favorite?
Our favorite digression?
Yes.
I think it has to be the weather because that's. And what is the weather?
By the way, it's overcast when I came in.
That's true. It's 16C and chance of rain.
16C, yeah.
People get very upset about those digressions.
Don't stop.
Okay.
I'm Mark Fuccio. I came in from San Jose, California. I have a comment and two questions. First comment, I've been listening to TWiV since around episode 80. And I want to thank Vincent for you and the panel for a decade or more of hard work. Let's have a round of applause. The second question is, we've seen recently the explosion of interest in machine learning. Now everything's called AI through ChatGPT, and there's other more specialized machine learnings that we see in photo apps on our phones and things like that. Are you aware of any interdisciplinary work applying machine learning techniques, for either advancement of viral research or development of drugs?
There's a lot going on with that, and there has been since before ChatGPT was a common topic of conversation, or before I'd even heard of it. People have been using machine learning extensively in analyzing huge data sets in genomics, and especially in medicine, where it's now possible with cooperation of groups like Kaiser Permanente, they can anonymize millions of medical records, and you can do these massive screens across huge numbers of people that were never possible in the past. And you need machine learning to do that kind of analysis because no human can possibly comprehend a data set that size. So yeah, that is very much an ongoing area.
Okay.
I have one follow-up question.
That's the last one.
I have a chronic TWiV listening habit. Is there no cure?
Ask Daniel.
There's an immunization. You need to get a weekly booster.
All right, that's it for the questions. Thank you, Nadia. Thank you. I think we could have made a whole show just on that. You guys are great. But I wanted to ask Brianne. You were on Jeopardy. How does that work, Brianne?
Well, let me tell you how it works. So we are going to play a little bit of TWiV Jeopardy today.
I'm in Jeopardy.
We need music.
So we have some questions prepared. There are 6 categories, each with 5 questions. And I will have you all raise your hand when you know what I'm talking about. Yeah, so please choose a category and amount. Raise your hand and answer in the form of a question.
Wait, who's playing?
So our categories
Are we
are.
going to invite an audience member just at random?
Yeah.
Yeah.
A lifeline. A lifeline.
Lifeline, and I'm sad I don't get to play. Our categories are parasites, RNA viruses, DNA viruses, viral ecology, TWiV immunity, or viral immunity, and TWiV trivia. So yes, Dixon.
Parasites for 200.
Yes. All right. This Drury University research fellow was awarded the Nobel Prize
Who was William C. Kennedy?
Ivermectin. Yeah. Raise your hand.
You gotta raise your hand.
Raise your hand.
Raise your hand.
And what's the answer?
Who is William C. Kennedy?
Correct. All right.
friend of mine.
We have an early lead here.
Yes. All right, so if we go for parasites for 400, we have, this is a colloquial name for Ascaris lumbricoides.
The giant, what is the giant intestinal worm?
Correct. All right, parasites for 600. This nematode parasite is commonly found in undercooked pork products. Yes.
What is trichinella?
Yes.
Should we add spiralis? Or is trichinella spiralis? And it could also be sarcocystis.
We have a contested answer.
All right. Parasites for 800. Parasites that migrate through human tissues as part of their life cycle are associated with this change in white blood cells. Daniel.
What is eosinophilia?
Correct. All right, parasites for 1,000. This cell type is formed from a muscle cell during trichinella infection. Angela.
The nerve cell?
Yes.
So what is the nerve cell?
Discovered by Dixon. All right, now we'll switch to RNA viruses. RNA viruses for 200. Antigenic variation of this virus necessitates frequent vaccine reformulations. Kathy.
What is influenza?
Correct. RNA viruses for 400. This RNA tumor virus was the subject of the longest incubation period for a Nobel Prize. Kathy.
What is Ralph sarcoma virus?
Correct.
Got the Nobel Prize when he was in his 80s. I lived long enough to
This
get
is
the
the
Nobel Prize.
DNA virologist is kicking our butts.
RNA viruses for 600. Amy Rosenfeld found extensive antibody cross-reactivity among members of this viral genus.
What is D68? No?
Genus.
Oh.
What are the coronaviruses? Oh, no, that's what's
Enteroviruses. Enteroviruses. Yes. You can have all of the magic beans. RNA viruses for 800. This one they actually asked me on the show. SARS-CoV-2 variant names skipped new and this 14th Greek letter. Daniel.
Oh, what did they skip?
Yeah.
So that was just.
They asked me this
Pi?
on actual record.
Is pi the 14th Greek letter? I don't
No.
know. Okay.
Something sounds like a Chinese president.
Yes, his side, because it sounds like she, yeah.
I'm not answering because I made up the question.
All right. RNA viruses for 1,000. This order of RNA viruses is the only whose members have a proofreading exonuclease. Kathy.
Oh.
Order.
What are the rhinoviruses?
Yes.
Is it you?
Somehow
I knew that.
All right. DNA viruses. This is the only human virus that has been eradicated. Rich.
What is smallpox?
DNA viruses for 400. Henrietta Lacks' cervical tumor was caused by this virus. Kathy.
What is HPV 18?
Correct. Very good.
Specific.
All right. DNA viruses for 600. The capsid of this virus contains 240 copies of hexon protein in addition to the hexon protein. Kathy.
What is adenovirus?
Correct.
Unfair question.
I wrote that one for you. DNA viruses 800. T antigens encoded in some DNA virus genomes must block the function of these two cell proteins to ensure the cells are divided. Kathy.
The two proteins are retinoblastine,
What?
PRB, and so what are PRB and P53?
Correct.
Good job.
DNA viruses for 1,000. The mythical jackalope is none other than a rabbit with papillomas caused by continuous expression of this protein.
You know this.
Got it, Angela? Go for it.
Is myxoma is P53? No. What is myxoma virus though, no?
Show fibrinovirus.
Oh,
What's the protein?
What's the
What's the protein? What is E6?
Yes. All right. Viral ecology for 200. There is evidence of this virus infecting white-tailed deer, allowing them to be possible reservoirs of variants that could potentially be transmitted back to humans. Daniel.
So what is SARS-CoV-2?
Correct. Viral ecology. At the Bronx Zoo, we're critical in understanding the emergence of this virus in the United States. Dixon.
What is the West Nile virus?
Correct. Viral ecology 600. This type of bat brought Nipah virus into piggeries in 1998 in Malaysia, leading to an outbreak among pigs and humans. Angela.
Is flying fox okay, or would you like
Flying fox
flying
is good. Viral ecology 800. Observations on this virus used as biological control of European rabbit populations in Australia are often cited as in discussion of changes of virulence over time. Rich.
What is myxoma?
Correct.
It came up again.
Yeah. All right. Viral ecology for 1000. This coccolithophore forms ocean blooms visible from outer space that are collapsed by viral infection.
I forget how to
Is
spell
it-
it.
Are you talking about the actual bioluminescence itself?
No, it's
Or is this,
the coccolithophore.
it's a dinoflagellate?
No.
What kind of?
All right, somebody knows in the audience. What is it?
Emiliana Huxley.
Yeehaw.
Yes.
Wow.
Yes. Emiliana Huxley. All right. Viral immunity for 200. This antibody appears first after infection or vaccination and then is replaced by IgG. Daniel.
What is IgM?
Correct.
You're actually excited to answer, aren't you? Love it.
All right, viral immunity for 400. This molecule presents peptides to CD4 T cells. Angela.
Well, do you say MHC? Or are we talking
What
about?
is
Sorry, what is the major histocompatibility complex?
Which one?
One.
For
Oh, for CD4 2?
Yeah.
Sorry. All cells have one though too. FYI.
Yes. All right, viral immunity for 600. This is probably the virus Edward Jenner used to inoculate James Phipps.
Well, that's... I'm going to go with what is horsepox.
Yes.
That help us.
Viral immunity for 800. This cytokine, discovered by Isaacs and Lindemann, induces the antiviral state and inhibits viral replication. Angela.
Interferon.
Yes.
Let's just say
You need to do the format. What is it?
I'm sorry, what is interferon? I've never played Jeopardy.
All right, viral immunity for 1000. This location in the lymph node is an important site for somatic hypermutation, affinity maturation, and memory B cell development. Daniel.
What is the germinal center?
Correct. That was good.
Someone keeping track of the points?
Yes.
All right, so now it's our last category, which is TWiV trivia. This was the topic of episode.
It was philosophy.
No.
West Nile virus.
Yes.
West Nile virus.
You were there.
All right, TWiV trivia for 400. This term for a portion of an antigen that is bound by an antibody is often used on TWiV as a malapropism for episode. Alan.
What is an epitope?
What is an epitope? TWiV trivia for 600. This guest joined an episode from the International Space Station. Kathy.
Who is Astro Kate? Kate Rubin.
Yes, Astro Kate Rubin. All right, 800, TWiV trivia. For episode 200, Vincent, Rich, and Alan toured this site. Yes, Alan.
What is the National Emerging Infectious Diseases Laboratory, or NEEDL?
That is correct. It is a needle. All right, our final question. TWiV trivia for 1000. Vincent says this tagline at the end of each episode.
It's the kind that make you sick. Actually, that's not it. No, that's not it. I screwed up.
This episode is...
Another TWiV is viral. So,
it goes.
Thank you, Brianne, that was great. That was fun. I think we should do more of that.
I have a lot of fun writing questions anytime.
All right, so that is TWiV Epitope 1000. Show notes are at microbe.tv/TWiV. You can send your questions and comment to TWiV at microbe.tv. If you enjoy our work, I know all of you do, but the others who are listening, please consider supporting us. We need your help to continue, and we'd like to work towards the next 1,000 episodes. Microbe.tv/contribute. This wonderful TWiV team, I think it's cool that they all came, besides you coming, right? It's great that they all came. Thank you for coming. Turbidplaque.com, Alan Dove.
Thank you.
Condon is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy. Is at BioProf Barker on Twitter, Drew University. Thanks, Drew. Daniel Griffin is at Columbia University Irving Medical Center and ParatitesWithoutBorders.com. Thank you. And Angela Mingarelli is from McGill University. Thank you, Angela. Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. I want to thank the Randall Desplombier Quartet for after this is over, we all go upstairs for a lovely reception. You can get to talk to us more, and they'll be entertaining us there as well. I also want to thank Ray Ortega, our producer there. Chris and Diane, our photographer. Both Ray and Chris I worked with for many years at the American Society for Microbiology live events, and in fact they were there for the first live epitope in Philadelphia where there was one person. So we've come a long way. Yeah, indeed. And thank you for coming and looking forward to your support for the next 1,000 epitopes. Now, as I said, join us upstairs for a reception. Before I sign off with my, what was it on the question? With my iconic greeting. Be seated after I sign off because Chris and Diane wants to get here a photograph of us with you in the background. So don't leave until that photo is over, all right? Okay. Got that? Okay. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.